Hi friends, welcome back to the class. Today I'm going to present a short video on extrinsic factors which are affecting the growth and survival of microorganisms in the food. Extrinsic factors, we will have three, that is relative humidity, temperature and gaseous atmosphere. These are the three extrinsic factors which have an impact on the growth and survival of the microorganisms. Coming to relative humidity, as I discussed earlier, relative humidity and water activity are interrelated. Thus, I can actually explain it as relative humidity is a measure of the water activity in the gaseous phase, right? So it is nothing but the water content in the air. So that's it is interrelated. This is measured by an instrument called hygrometer. This we have been studying from our young ages. Then when food commodities having low water activity are stored in an atmosphere of high relative humidity, water will transfer from the gaseous phase to the food. So that is how condensation may occur on the surface, giving rise to localized regions of high water activity. That is what I have tried to make it in the PPT. In this situation, the propagules which are viable now will germinate and grow, which are not viable will not, but some viable propagules will be there which will be waiting for a suitable conditions to grow. So microorganisms produce water as an end product of respiration. We should all know that. This increases the water activity of their immediate environment, making way for other microorganisms to grow. So this type of atmosphere wherein the respiration gives the water, and this makes the environment suitable for other organisms which require higher water activity to grow, and then that causes the spoilage of a food. Okay, so a classical example for this is the storage of grains in silos. You might have seen the big silos which are actually erected outside the dairy. If you have visited dairy, milk dairies, you can see. Or you can actually take it as concentrated syrup in the tanks also in the juice making plants and all. What happens here is the small explanation here in this scenario is the relative humidity of the air is very sensitive to temperature. If the temperature rises at the daytime, heating up only one side of the silo or the tank, whatever you call it as, there is a net migration of water molecules inside to equilibrate the relative humidity. But when the same side gets cooled down during the night or evening, when the sun is not at that temperature, water activity becomes more for a shorter period before the water molecules migrate back. This short period is sufficient to cause local condensation. So, this allows germination of fungal spores and subsequent spoilage of the grain. These all happens because they are very huge and they are directly exposed to the sunlight. They cannot be housed inside the building. Okay, the storage tanks and silos. It is of course housed inside the campus, inside the compound, but it is quite exposed. So if you go to the dairy, you can see that. So this is what happens in the when the equilibrium of the relative humidity is disturbed. This is what happens, and this much a fraction of a second or few minutes is enough for the organisms to activate. So that is about your relative humidity. Passing on to temperature. Temperature, you all know that this is one of the most important factor, and microbes can grow right from -8 degree to 100 degree centigrade, of course with the exceptional cases. Each organism will have cardinal temperatures. Cardinal temperatures means it has a minimum, optimum and a maximum temperature at which it can grow best or worst or anything. Based on these cardinal temperatures, the microorganisms can be classified into several physiological groups. I have made a tabular column and I have tried to include all the different physiological groups, that is psychrophiles, psychrotrophs, mesophiles, and thermophiles. There is, because many of you will not know the difference between psychrophiles and psychrotrophs, I would like to say that there is a difference between psychrophiles and psychrotrophs. Minimum temperature for the psychrophiles being -5 to +5. Optimum is 12 to 15. Maximum is 15 to 20. And all these Pseudomonas type of microorganisms can be grown. This you will see in all your refrigerators, right? Psychotropes means, of course, minimum temperature will be same as -5 to +5, but the optimum temperature will be 25 to 30. That means when the weather is not so hot, even at the colder weather outside the refrigerator also, this temperature persists, that is 25 to 30 prevails, and then you can see the psychotropes. Maximum will be 30 to 35, almost nearing your mesophiles. So examples are Cladosporium, Penicillium, Listeria, Clostridium, Botulinum. All these are examples. And I can call these psychotropes as facultative, that is both. It can grow in that psychrophilic conditions also as well as above that. Then psychrophiles as obligate. Okay, so they require compulsorily the set temperatures. Coming to mesophiles, mesophiles will have 5 to 15 as minimum temperature, 30 to 40 as an optimal temperature, a very suitable environment, and 40 to 47 as the maximum. It can withstand till 47. So the examples being Bacillus, Salmonella, Aspergillus, Staphylococcus, all these are, you have a variety, a very wide range of mesophilic microorganisms. So you can name all those. I have just named few. Coming to thermophiles, you all know they are a heat loving organism. Then 40 to 45 will be the minimum temperature. Optimum will be 55 to 75, and above that is maximum, that is 60 to 90. So till 85, 90, 85, 90 it can withstand. And a suitable example is Bacillus, Clostridium, all this. So these physiological groups has to be remembered when you are discussing or when you are actually guessing the type of microorganisms in the food. It is very clearly evident from this graph, what I have written, that the curve is asymmetric. That is, the growth declines more rapidly above the optimum temperature than below it. Previous to that, you can see a gradual increase. It is not a slide like that, but after the optimum temperature is done with that, there is a sudden fall of the graph, right? So rapidly above the optimum temperature than below it, I have told. As the temperature is decreased from the optimum, the growth rate slows, partly as a result of the slowing of enzymic reactions. I'm sorry, it got expanded. Okay, so as the temperature increases above the optima, the growth declines more sharply as a result of irreversible denaturation of proteins and the thermal breakdown of the cell's plasma membrane. So once the proteins are denatured, you cannot get back the proteins into the normal condition. So growth of organism at different temperature is also dependent upon other factors such as water activity, pH, and nutrients. Coming to gaseous atmosphere, various gases too have an impact on the growth and survival of microorganisms. Many microorganisms have specific oxygen requirement for growth that are used to categorize them into four different groups. You know, aerobic, anaerobic, facultative, obligate. You have studied that in previous factors. Oxygen, you all know, that comprises 21% of the Earth's atmosphere. Its presence and its influence on redox potential are very important determinants of the microbial association and their rate of growth. The inhibitory effect of carbon dioxide on microbial growth is applied in the modified atmosphere packaging. MAP means modified atmosphere packaging, and CAP is controlled atmosphere packaging. In these types of packaging, we use carbon dioxide and we take an advantage of its nature in the food and the temperature. Fine. Molds and gram-negative organisms are more sensitive to when compared to gram-positive bacteria. So these gram-positive bacteria tend to be more resistant. Brettanomyces species and yeast will show tolerance to high carbon dioxide level. So what happens with the carbon dioxide? The growth inhibition is greater under aerobic conditions than anaerobic conditions. Inhibitory effect increases with decrease in temperature due to increased solubility. Okay. So what happens in gaseous atmosphere? What is the sorry role of carbon dioxide on pH? We will see. Effect of carbon dioxide on pH. Carbon dioxide dissolves in water, giving carbonic acid. So this is a weak acid, and this partially dissociates into bicarbonate ions and protons. So this we have studied in pH. So thereby lowering the pH values. Other contributory factors are thought to include changes in the physical properties of plasma membrane, adversely affecting the solute transport. They also inhibit the key enzymes, particularly those involving carboxylation or decarboxylation reactions, in which carbon dioxide is mainly captured. So each of these compositions, each of these compositional factors of food, both intrinsic as well as extrinsic, can significantly affect the resultant microbial flora. Many of these factors interact, and therefore we should be concerned about the total ecology of the food. The difficulty of collecting numerous data points over a wide range of variables may result in the need of a constructive mathematical models to predict a suitable preservation system. This is nothing but your predictive microbiology, predictive models. All these have come into existence now. We have predictive models which is predicting the status of the particular microorganism in a particular specific food. So n number of foods, n number of microorganisms have a prediction called predictive models, and that by that the consumer who buys the food can be very clearly explained what is the fate of the organisms or what is the fate of this particular food over the period of time. So using this we can decide whether to use this food or not. This is about the extrinsic and intrinsic factors which affect the growth of microorganisms. Coming to implicit factors, I had told about the antagonism, that is bacteriocin against bacterial species, bacteriocin against yeast by cell membrane. All these things have been dealt with in your previous classes where you study antagonism as a factor. Agglutinate and aggregation and agglutination of the organisms. Enterobacteriaceae and lactic acid bacteria will agglutinate. Even Saccharomyces by reaction with surface nanoproteins of yeast, which will happen. Commensalism is also one great factors. Complex proteins and carbohydrates are degraded by some species of some species to make it a simple carbohydrate or proteins. Organic acids are cut down by or broken down by yeast and molds that will also favor the bacterial growth. And also one thing, bacteriophages, which will destroy the starter cultures of lactic acid bacteria in fermentation process, also to be considered. Phages against Pseudomonas and Leuconostoc, Propionibacteria in cheese, Leuconostoc in wines, Pseudomonas in refrigerated meat. All these play an important role as implicit factors in determining the factors affecting the growth and survival of microorganisms. So I think I'm clear about the factors now. We will move on to the next chapter. Till then, please review the factors, which are all the causes. This can be asked as a single question for the 15 marks. Also, all the factors can be asked as short notes as well as 5 marks questions. Thank you for your patience listening. Bye bye.